Somewhere in the Kalahari Desert, in a dry expanse of sand and thornbush stretching across Namibia and Botswana, there are people alive today whose DNA carries a message that no other population on Earth can deliver. Scientists who have sequenced their genomes describe what they find with the kind of careful, measured language that researchers use when they know this is where the story of human genetic age begins. And it begins with a question that sounds simple until you actually try to answer it. How do you measure the age of a population? Not when a culture began, not when a language first formed, not when a group settled a particular piece of land, but when genetically a group of people first separated from all other humans and began accumulating the specific mutations that now define them as distinct. That question, which once seemed impossible to answer, is now precisely what modern genomic science attempts. And the answers, in case after case, have shocked the researchers who found them. The tool scientists use is the haplogroup. A haplogroup is a cluster of genetic variants inherited together, passed down, unchanged, from parent to child across generation after generation. Mitochondrial DNA passes only through mothers. The Y chromosome passes only through fathers. Both accumulate small mutations at predictable rates, acting as what geneticists call blood. When researchers at the J. Craig Venter Institute and Penn State University published the first complete genome sequences of Khoisan individuals in the journal Nature in 2010, they reported something that immediately reframed the conversation about human origins. The Khoisan people of southern Africa represent what geneticists describe as the oldest known lineage of genomes across 48 ethnic groups worldwide, that the Khoisan were genetically distinct not only from Europeans and Asians, but from all other Africans as well. They represent a lineage that separated from the rest of humanity's gene pool so long ago that no other living population today carries equivalent depth. Their roots in the genetic record stretch back, depending on the specific comparison. When most people think of ancient populations, they think of isolated island tribes, of remote mountain communities, of groups in the deepest jungles of South America. They do not think of a group of people living in a southern African desert who speak with clicking consonants and whose rock art covers cave walls going back tens of thousands of years. Yet the DNA does not care about expectations. Research published in Nature Communications, analyzing genetic data across 162 genomes from 48 ethnic groups, concluded that the Khoisan were once the largest population of modern humans on the planet, comprising the majority of all living humans for much of the past 150,000 years, before a decline began roughly 22,000 years ago. That is an extraordinary reversal of what most people assume about human history. The populations we tend to think of as dominant, the great agricultural civilizations of Eurasia, the builders of empires, the ancestors of the world's majority populations today, are, genetically speaking, recent arrivals. Their lineages are younger, their haplogroups newer, their divergence from a common ancestor relatively shallow. The Khoisan, by contrast, are carrying a genetic passport stamped at the very beginning of human history. In 2013, a genetics paper published in the American Journal of Human Genetics sent a quiet shockwave through the scientific community. It did not make front-page news immediately. It did not arrive with a press conference or a dramatic announcement. It came from a laboratory database, from a citizen genealogy project, and from the Y chromosome of a man who had been dead for well over a century. When the results were confirmed, the researchers involved understood they were not simply looking at an unusual genetic variant. They were looking at the oldest known paternal lineage ever identified in a living human, a lineage so ancient it forced scientists to completely recalculate when all men alive today shared a common male ancestor. The story begins with a woman named Jacqueline Johnson, who submitted a DNA sample from a male cousin to the African American DNA Roots Project, a citizen science initiative designed to help African Americans trace ancestry that slavery had erased. The male cousin's Y chromosome did not match anything in the existing database. It did not fit any known haplogroup. It sat outside the entire established structure of the human paternal family tree, at a position so deep that geneticists had not previously mapped it. When researchers at the University of Arizona, led by Fernando Mendez and Michael Hammer, analyzed the sample, they traced the male line back to a formerly enslaved man from South Carolina named Albert Perry. The discovery, published in 2013, revealed that Perry carried what geneticists designated Haplogroup A00, the oldest known male lineage ever identified, dating back, by initial estimates, more than 300,000 years. Paintings, before the first evidence of symbolic thinking, before modern humans had reached Europe or Asia. The extreme genetic divergence of haplogroup A00 forced a radical recalculation of the molecular clock. The initial 2013 analysis, incorporating A00 at the base of the Y chromosome tree, suggested an age for Y chromosomal atom of approximately 338,000 years. a figure that pushed the estimated root of the male family tree back further than the emergence of anatomically modern Homo sapiens, as scientists had previously defined the species. That last part demands a pause. The estimated age of Haplogroup A00 is older than the oldest commonly accepted fossils of anatomically modern humans. The bones found at Jebel Irhoud in Morocco, which currently represent the earliest known Homo sapiens remains, date to approximately 300,000 years ago. A paternal lineage older than those fossils introduces a possibility that made researchers deeply uncomfortable. Either the molecular clock used to estimate mutation rates was off, or the ancestors of this lineage were part of a human population that existed, reproduced, and then largely disappeared, leaving behind only this faint genetic echo in the descendants of an enslaved man in South Carolina and in a small population of men in Western Cameroon. When researchers compared Albert Perry's Y chromosome markers to genetic databases, they found a match among 11 individuals out of 174 sampled from the Mbio people, a Bantu-speaking population in Western Cameroon. The same ancient lineage, dormant and unrecognized in one population for millennia, had survived on two continents simultaneously, in West Central Africa, where it likely persisted in a small isolated community, and in the American South, carried there against all by the machinery of the slave trade. The haplogroup that rewrote the timeline of human paternal ancestry was not found through a major funded archaeological project. It was found through a genealogy kit and a woman trying to trace her family. But the story of genetic age is not solely written by outliers. The broader pattern, when researchers examine populations around the world, reveals something consistent and counterintuitive. The populations that score as genetically oldest, measured by the depth of their divergence from other groups, by the range of their internal diversity, by the age of their founding haplogroups, are overwhelmingly populations that modern history has treated as marginal. They are hunter-gatherers. They are small island communities. They are groups that did not build empires, that did not expand across continents in agricultural waves, that did not replace other populations through conquest. Genome-wide analyses suggest that ancient African populations began taking on genetic structure around 200,000 years ago, with a divergence between Khoisan and non-hunter-gatherer African groups occurring by approximately 160,000 years ago, a split that predates the founding of virtually every other major human population lineage outside Africa. What that means, in practical terms, is that the deepest roots of the human family tree are not found in the civilizations that wrote history down. They are found in the people history largely ignored or actively tried to erase, the San of the Kalahari, the Embo of Cameroon, the isolated communities whose genetic signatures were only discovered because scientists looked carefully enough. And then there is another group, separated from the rest of humanity, not by the Sahara or the Congo Basin, but by 60,000 years of open ocean. In the Bay of Bengal, roughly 3,300 kilometers off the eastern coast of India, there's a chain of islands where some of the oldest surviving human lineages outside Africa still exist. The Andaman Islands rise from the Indian Ocean in dense jungle, ringed by coral reefs, cut off from the mainland by deep water that, for most of human history, was effectively uncrossable. The people who live in the most protected interior zones of these islands, the Garawa, the Onge, the Sentinelese, are not simply isolated in the modern political sense. They are isolated in a genetic sense that goes back to a moment near the very beginning of human expansion out of Africa, when a group of early migrants followed the coastline eastward and eventually found these islands, settled them, and were then cut off from the rest of the human story by rising sea levels approximately 10,000 years ago. Research supports that the Jarawa people are likely descendants of the first humans who left Africa approximately 60,000 to 70,000 years ago, traveling along coastlines through the Arabian Peninsula and into Southeast Asia before reaching the Andaman Islands, where isolation subsequently preserved their genetic distinctiveness for tens of thousands of years. What makes the Andamanese genetically extraordinary is not simply the age of their lineage, but the completeness of their separation. While the Khoisan and the carriers of Haplogroup A00 represent the deepest roots of human diversity within the African continent, the Andamanese represent something different, the oldest known surviving branch of the migration that eventually populated the entire rest of the world outside Africa. Every person alive today in Europe, Asia, the Americas, and Oceania descends from that original out-of-Africa movement. The Andamanese separated from that stream near its very beginning and then stopped moving. When researchers analyzed mitochondrial DNA and Y chromosome markers from the Onge, Jarawa, and Great Andamanese tribes, they found genetic variability consistent with long reproductive isolation, concluding that the Andamanese represent descendants of the early Paleolithic colonizers of Southeast Asia, with their mtDNA lineages having survived in complete genetic isolation from other South and Southeast Asian populations since the initial settlement of the region following the out-of-Africa migration. In the language of population genetics, these communities exist at a branching point so early in the human dispersal record that their continued existence is scientifically remarkable. Most populations that separated from the mainstream of human migration this long ago have since been absorbed, replaced, or admixed into subsequent waves of movement. The Andamanese, protected by ocean and by choice, were not. What researchers found when they studied the specific haplogroups carried by Andamanese populations reinforced this picture. Their mitochondrial lineages belong to branches of the macrohaplogroup M, an ancient East African-derived line that represents one of the two founding haplogroups of the entire out-of-Africa dispersal, the other being haplogroup N. Macrohaplogroup M is estimated to have arisen somewhere between 60,000 and 75,000 years ago, shortly after the first successful migration out of Africa. The Andamanese carry variants of M that diverged from other M lineages very early, before the subsequent waves of migration that would eventually colonize South Asia, East Asia, and Oceania. Their DNA is, in effect, a direct line back to the founding moment of the human diaspora. But the picture of genetic age across global populations is not simply a story of which group is oldest. It is a story of what happens to populations over time. Specifically, what happens to their genetic diversity when they expand rapidly, versus when they remain isolated and small. This is where a concept called the founder effect becomes essential to understanding why the world's populations look genetically the way they do. When a small group of people migrates away from a larger population and establishes a new community, they carry with them only a subset of the original genetic diversity. Every subsequent migration compounds this narrowing. The populations of Europe, East Asia, and the Americas all passed through successive genetic bottlenecks as their ancestors moved further and further from the original African source population, each time taking a reduced sample of the full human genetic range. The consequence of this is that the populations most distant from Africa, not geographically distant as we experience distance today, but most removed from the original source through successive migrations and founder events, tend to carry the least internal genetic diversity. Research on ancient DNA has progressively revealed the complexity of the admixture. Landscape involving European hunter-gatherers, Anatolian farmers, and Western Eurasian steppe pastoralists, with modern European populations being the product of at least 3 major ancestral streams mixing within the last 10,000 years. What this means is that the populations that built the great historical civilizations of Eurasia are, in genetic terms, relatively recent composites, populations assembled from the mixing of other populations over the past few thousand years, not ancient singular lineages stretching back to human origins. The populations that score as genetically deepest, by contrast, are precisely those that did not participate in the great agricultural expansions of the Holocene. They are the groups that stayed small, stayed isolated, and kept accumulating their own internal diversity without absorbing large waves of outside migrants. Their genetic age is not a measure of superiority. It is a measure of stillness, of what happens when a population remains in one place and relatively stable conditions for long enough that time itself writes its signature into their genome in ways no other living group can match. In remote island communities, but in a database, in an ordinary modern genome, in a man whose descendants had not the slightest idea what they were carrying. There is a question that emerges naturally from everything the science has established, and it is the question that the researchers themselves keep returning to. If the oldest genetic lineages belong to populations that stayed small, that remained isolated, that did not expand outward in great agricultural waves, what does that actually tell us about the nature of human history? Not about which group is superior, not about which culture achieved more, but about the specific, verifiable mechanism by which genetic age is created and preserved. Because the answer to that question turns out to reveal something profound about the relationship between human diversity and human survival, something that has direct implications for medicine, for anthropology, and for how the entire species understands its own origins. The key insight is this. Genetic age, as scientists measure it, is fundamentally a measure of retained diversity. Much of the past 200,000 years of human evolution has been a story of population structuring and diffusion within Africa, with deep splits between ancient groups producing the extraordinary range of genetic variation still visible today in Africans. Populations, variation that dwarfs anything found in populations outside the continent. When researchers compare the total genetic diversity found across all non-African populations, combined against the diversity found within Africa alone, Africa wins. Not by a small margin. The entire genetic range of humanity outside Africa represents a subset of the diversity that exists within Africa. Every European, every East Asian, every indigenous American, every Pacific Islander traces their ancestry to a group that left Africa carrying a limited genetic sample, and every subsequent migration further narrowed that sample. This is why the ranking of populations by genetic age is not, at its core, a ranking of populations by any quality we would ordinarily think of as significant. It is a ranking by proximity to the source. The Khoisan of southern Africa were found to be the majority population of living humans for most of the past 150,000 years, with their genetic diversity reflecting the full depth of that ancient demographic dominance. A dominance that collapsed not because of any failure of their own, but because other populations expanded outward explosively while the Khoisan remained in place. Their genetic richness is the direct product of that stability. They did not lose diversity through bottlenecks. They did not have their gene pool narrowed by rapid migration. They accumulated time, and time gave them depth. The broader implications of this understanding have begun reshaping how medical researchers approach human health. For decades, the majority of large-scale genomic studies used European populations as their primary reference. The Human Genome Project itself drew heavily from a small number of donors, most of whom were of European descent. The consequence of this bias is that the genetic variants associated with disease risk, drug response, and inherited conditions are far better characterized in populations of European ancestry than in anyone else. The genetic structure of the indigenous hunter-gatherer peoples of southern Africa is important not only for understanding human diversity, but for facilitating the inclusion of southern Africans in medical genomics research, a gap that has left their populations underserved by the very science that their genomes could most enrich. The populations that are genetically oldest, that carry the deepest diversity, that represent the widest range of human variation, are precisely the populations that medical genomics has historically studied least. What researchers like those at the Max Planck Institute for Evolutionary Anthropology have argued is that this is not merely an ethical oversight. It is a scientific one. Research into the genomes of the earliest modern humans shows that after leaving Africa, populations met and interbred with archaic groups like Neanderthals, resulting in roughly 2 to 3% Neanderthal DNA visible in all people outside Africa today. Yet the full picture of what ancient African populations interbred with and what those interactions contributed to modern human biology remains far less understood precisely because African genomes have been studied less comprehensively. The oldest populations are carrying answers to questions that science has not yet fully learned to ask. The case of Haplogroup A00 pushes this point to its furthest edge. The discovery that the A00 lineage predated all known haplogroup A branches forced a recalculation of Y chromosomal Adam's age, with researchers considering two primary interpretations. Either this represented a long isolated population of early Homo sapiens in Africa that remained genetically distinct, or the Y chromosome may have been introduced into the modern human gene pool through interbreeding with an unknown archaic hominin population, a hypothesis that remains actively debated. Either interpretation is extraordinary. Either a population of modern humans lived in near complete isolation for hundreds of thousands of years, maintaining a distinct genetic lineage that eventually fed back into the broader human family through a single South Carolina man's ancestors, or our species interbred with an archaic relative in Africa, whose identity we have not yet confirmed, in a meeting that left a genetic trace now detectable in living descendants. Both possibilities expand what we thought we knew about human history into territory that feels almost impossible. What the full body of this research leaves behind is not a simple ranking. It is not a table in which populations are assigned scores, and one group declared the winner of human antiquity. It is something more complicated and more honest than that. The populations whose DNA carries the deepest time signatures are populations that survived, not through expansion, not through conquest, not through the accumulation of territory, but through persistence. The San of the Kalahari, hunting across the same landscapes their ancestors crossed 150,000 years ago. The Jarawa of the Andaman Islands, living within the same forest system their founding ancestors entered after crossing an ocean 60,000 years ago. The descendants of Albert Perry, carrying without knowing it a Y chromosome that science had not yet invented the tools to recognize. The question of which population is oldest is, in the end, a question that collapses into a larger and more important one. What has all of this time produced? The answer is diversity. A range of human biological variation so vast, so layered, so irreplaceable, that the populations currently least studied by science are precisely the populations most urgently worth understanding. Every ancient lineage that disappears through assimilation, disease, or displacement takes with it genetic information that accumulated across time scales no modern institution could ever reconstruct. The molecular clock keeps running, but it only preserves what survives. The oldest genomes on Earth are not fossils. They are living people. And the science that is finally learning to read them properly is only beginning to understand what they have been carrying all along.